Namaskar, Davyon or Sajanon. Imagine a vast library containing every instruction to build a living being. That's DNA. DNA, or deoxyribonucleic acid, is life's blueprint, holding the code for everything from your eye color to your heartbeat. Every time a cell divides, this entire library must be copied perfectly. This is DNA replication. If mistakes happen, the consequences can be severe, so precision is vital. Replication is the engine of continuity, passing instructions from one generation to the next. But how does this copying actually happen? Is the original kept safe, or is it split and used as a template? Scientists needed a way to peek inside the cell and witness this process. The quest to solve this mystery led to a discovery that changed our understanding of life. To understand replication, we must first see DNA's structure. A twisted ladder, the double helix. The sides are sugar phosphate backbones. The rungs are pairs of 4 bases: adenine, thymine, guanine, and cytosine. A always pairs with T, G with C. This strict pairing makes the strands complementary. Knowing one side's sequence reveals the other, ensuring reliable information storage. The sequence of these base pairs forms the genetic code, instructing cells what proteins to make. Watson and Crick's discovery of the double helix suggested a copying mechanism, unzipping the ladder, each strand serving as a template. If each strand guides the creation of its partner, DNA could be copied with remarkable accuracy. This was a beautiful idea, but it needed proof. Only a clever experiment could reveal the truth. Scientists proposed 3 models for DNA replication. Conservative, the original helix stays intact, and a new copy is made from scratch. Dispersive, DNA is cut into pieces, and each new molecule is a patchwork of old and new. Semi-conservative, the helix unzips, each strand serving as a template for a new partner. The semi-conservative model seemed logical, but evidence was needed. The challenge: how to distinguish old DNA from new after replication. This puzzle set the stage for a groundbreaking experiment. Enter Messelson and Stoll. In 1958, Messelson and Stoll designed a brilliant experiment. They grew bacteria in heavy nitrogen, N15, making all DNA heavy. Then, they moved the bacteria to light nitrogen, N14, and let them replicate once. Using density gradient centrifugation, they separated DNA by weight. After one replication, DNA formed a single band at an intermediate density. Not heavy, not light. This result ruled out conservative replication, which would have produced 2 bands. The intermediate band suggested each DNA molecule was a hybrid of old and new. But was it semi-conservative or dispersive? They let the bacteria replicate again in light nitrogen. The answer was about to be revealed. After a second replication, 2 bands appeared. One intermediate, one light. This matched the semi-conservative model. Each DNA had 1 old and 1 new strand. Dispersive replication would have produced only a single, gradually lighter band. Messelson and Stoll's experiment proved DNA replicates semi-conservatively. This discovery explained how genetic information is faithfully passed on. It's the foundation for modern genetics, medicine, and biotechnology. Understanding DNA replication helps us fight diseases, develop therapies, and solve crimes. Every advance in genetic science builds on this elegant experiment. The story of how life copies itself is now part of our shared knowledge.